Welcome again to our Age Art event at the Max-Planck-Institut for Biology of Aging in Cologne. I am standing here in our auditorium, you can see it here behind me, where we have space for almost 200 people. Tonight we have two special guests, Professor Janet Lord and double bassist Stanislav Anischenko. Professor Lord is Professor of Immune Cell Biology, and she's also Director of the Institute for Inflammation and Aging at the University of Birmingham. Her research focus is in the effect of aging upon immune functions. And tonight she will present her latest work related to COVID-19. She will also talk a little bit about statins, cholesterol medication, and what they have to do with fighting COVID-19. Yeah, this you will hear in just a moment. For the second part of our presentation, we have a special guest from the WDR Symphony Orchestra here in Cologne. Double bassist Stanislav Anischenko is one of the most renowned artists of our generation. He is most known for his expressive power and his virtuosity. Tonight, he will play solo pieces written especially for the bass. A number of research studies have shown that both performing and listening to music can indeed have a positive impact on our immune system. The COVID pandemic has affected us all in some way, and we try to cope with it using different strategies. What better way to do this than through the powers of music? Thank you for joining us tonight, and I hope you will enjoy the presentation.
Hello, and it's my great pleasure to be speaking at this Age Art event, combining science and art in the same lecture. Really unusual, and looking forward to the music as well involved. What I'm going to speak about today is SARS-CoV-2, the coronavirus, and the disease it causes, Covid-19, and how this virus that has no eyes can see the older adult with devastating consequences. I'm going to take you through some of the features of the immune system, what happens to it with age, and why this makes older adults more susceptible to this infection. So, if we begin, as I said, I'm going to cover the virus itself, tell you a little bit about the virus. I'm going to go through demographics, how our immune system deals with viruses, why older adults are more susceptible, and I'm going to try and finish on a positive note about vaccines and how they may help us out of this current pandemic. So first of all, the coronavirus, SARS-CoV-2. All viruses are very simple structures. They're essentially a single cell with a protein on the outside, and this protein is how they dock onto the cells in our body. Think of it a little bit like a lock and a key. In the case of the coronavirus, SARS-CoV-2, this is called the spike protein. Inside it has its genetic material, and it has to bind onto the cells in our body to take its own genetic material inside so that it can replicate. These viruses cannot replicate or survive outside of the body. In the case of SARS-CoV-2, this genetic material is RNA. The receptor, the lock that it binds to, is called the ACE2 receptor, and this is found primarily on the cells in our airways, so our mouth, our nose, and our lungs, although it is also in our intestinal tract. So this is why it's a respiratory virus, because it can only infect those cells. So what about these demographics then? These are quite stark. These are data from the UK, but it's pretty well the same everywhere, in Germany as well. If you look at the number of people and their age who succumb to this infection, you can see it's really age-related. Males on the left, females on the right, and over 90% of the cases are actually in the over 65-year-olds. Younger adults, of course, get it too, but they're asymptomatic, so we don't see them in the hospitals most of the time. And what about the severity of the disease? On the left here, I've got the data for those patients who only require a low level of care, so they don't get ventilated, they don't end up in intensive care. And again, you can see it's very age-related. The older you are, the chances are you will end up in hospital. On the right are the patients who end up in critical care, and again, highly age-related. The older you are, the sicker you will be with this virus. And lastly, the chances of you passing away with this virus are largely age-related. So most of the deaths, again, are in the over 85-year-olds. So why is this? To try and understand, you first need to understand a little bit about how our immune system responds to viruses and, well, any pathogen. So this video from the British Society for Immunology will hopefully help you to understand that and the rest of my talk.
The human immune system is composed of a complex network of immune cells, particularly white blood cells, also known as leukocytes. These have many different roles, but ultimately they work together to keep the body safe from potential danger, like a microscopic personal emergency services team. Sources of danger include invasion by pathogens, like harmful bacteria, viruses, fungi, or parasites. The immune system protects us using two approaches, known as the innate and adaptive immune responses. The innate immune system acts first and is made up of emergency first responder cells, signals, and antimicrobial substances, aimed at removing anything which looks harmful. The adaptive immune system consists of specialized cells and their products, which are trained over time to neutralize a specific threat. The innate immune system doesn't require any previous information about the threat, but its less focused attacks can cause more damage to nearby cells. The adaptive immune system is very specifically targeted to a pathogen, but needs prior information about the invader and time to set up an effective response. The exact details of how the new coronavirus triggers our immune system are now emerging, thanks to ongoing medical research. The ability of immunologists to discover these details is vital to understanding the natural development of protection and to create the best vaccines, as well as safe and effective drugs to treat different stages of COVID-19. We know that when the virus first enters the body, it inserts its genetic material into the epithelial cells, those cells lining the upper airways. This hijacks those cells to produce and assemble all of the components required to make copies of the virus. Those hijacked infected cells will eventually die, releasing millions of new viral particles, which start the cycle again. In somebody that's never encountered the virus before, the innate immune system responds first. Leukocytes called macrophages, living in and monitoring these infected areas, eat up bits of virus, the dead cell. Alerted to the problem, the macrophages release alarm signal proteins called cytokines, which bring other leukocytes to the scene, including neutrophils and natural killer cells. Inflamed tissue starts to populate with these cells, ready to act. Together, these different types of leukocyte can try to destroy infected cells and prevent viral spread. Epithelial cells can act too, making proteins which sound an alarm, preventing viral entry, or protecting neighboring cells by directly damaging the virus. In the best case scenario, this innate immune response may be able to clear the virus before more significant disease occurs. However, if these responses are not properly regulated and inflammation becomes uncontrolled, this process can actually start to damage the body. While the innate immune system tries to deal promptly with the viral infection, other immune cells called dendritic cells and macrophages start to train the adaptive immune system by showing bits of the virus to T and B cells. There are millions of these T and B cells inside your body with unique proteins called receptors on their surface. If one of these cells has a receptor that fits with a bit of the virus, this cell gets selected and activated. Once those best fit T and B cells have been identified, they multiply to make clone armies of themselves, all trained to fight this specific threat. Some of the trained T cells identify and kill lining cells infected with the virus. This helps to remove the infection, but needs to be carefully controlled so that it doesn't do too much damage. Other T cells send signals that regulate the body's responses and also help B cells to activate. B cells are unique in that they can make antibodies. These protein tags will stick to the virus. Neutralizing antibodies prevent the virus from entering cells and can help other cells to remove and destroy the viral particles. However, other antibodies can stick to the virus without doing anything of great value. And rarely, some antibodies may even help the virus to enter our cells. Neutralizing antibodies are often the best weapon against viruses, and immunologists are working to understand if these might also protect us against COVID-19. If the innate and adaptive immune responses manage to neutralize the virus, they start to release new signals, which will calm the inflammation, encourage healing of the tissue, and return the immune cells to a monitoring state. These people might have had almost no symptoms of COVID-19, but the adaptive immune system might still form protective memory against future encounters with the virus.
Hopefully that's now giving you an introduction so that you have a little bit of an understanding of how the immune response works against the virus. What I'm going to introduce you to now is essentially what goes wrong and why COVID-19 develops. What we know so far is when you look at patients who've developed severe COVID-19, if we look in their lungs, what we find is a lot of tissue damage. And you will have heard in that video that sometimes these first responder cells, the macrophages and the neutrophils, unfortunately do tissue damage as well when they're trying to clear the infection. And what seems to happen in patients who get severe COVID-19 is that they have an excess of these cells in their lungs. More importantly, when you look at people who do well and you collect cells from their lungs, they have actually got those adaptive cells, the ones that are trained specifically to kill the virus. They have those in their lungs and less of the macrophages. So it seems as though those who get severe COVID-19, their immune system really doesn't progress from that innate to the adaptive phase, and they get stuck in that rather harmful, damaging state. So why is that? Well, in the time I've got allotted, I can't go through everything, but I'm going to introduce you to one of the cells involved, the neutrophil. It's one of these fast responder cells. It's the most dominant white blood cell in your blood. You literally have hundreds and thousands of these cells. And how they do their job is that they go around in the blood, and when you've got an infection, they receive signals, those cytokines, from where the infection is, and that guides them towards the infection. It's like a signal for them. And this process is called chemotaxis. So they move from the blood to wherever the infection is. In the case of SARS-CoV-2, that's the lungs. When they get there, they literally then eat the target. So that's the neutrophil there, and this is a bacteria that it's ingesting. This process is called phagocytosis. Once the pathogen is inside, it then kills it by making chemicals such as reactive oxygen species. What I'm going to introduce you to now is what happens to as we get older, and to help you understand why COVID-19 is so much more severe in older adults. And I'm going to focus on this process of chemotaxis. So here's what it looks like. What we do here is we take a neutrophil from a person's blood, we put it under the microscope, but we give it also the target, so the bacteria or the virus here. And you can see here, so the neutrophil has started here, it moves up, and we've tracked it using a video camera so that we can see how well the neutrophil moves, what speed it goes at, if it goes in the right direction. And these are neutrophils from a young person. And you can see they're going beautifully straight lines, and these cells move very, very quickly. So we usually only have to record for about 9 or 10 minutes. What happens in an older person? This is an older person's neutrophils. And I don't think you need to be a professor of immunology to see that there's a problem here. The target is at the top of the screen here, but this one's going sideways, this one's going backwards. Hopefully you can see they wriggle around a lot. I always say it looks like they've lost their GPS system. These cells just don't know where to go. So they can move well, just not in the right direction. And here's the analysis. This is where we've just looked at how quickly the cells moved in a young and an old person. There's no problem there, the cells can move. This is where we've looked at this chemotaxis, movement in the right direction. And this is about 50% down as we get older. Why does that matter? Well, it might take the neutrophil longer to get to the site of infection, but we know that's not the main issue. When you look in the lungs of patients with COVID-19, they've got lots of neutrophils there, so they can get there. The problem is how they get there. This is a picture, this is a blood vessel, and this in the middle is a neutrophil. What they do when they leave the blood is they literally squeeze across the blood vessels to get into your tissues. And they release enzymes to digest the tissue. Literally, imagine them rather like a worm burrowing through soil. So they cause damage when they're moving. So your immune system inadvertently can cause damage, and this causes more inflammation in your tissue, which makes you sicker. And we calculated that if we were to compare the migration of an old neutrophil with a young neutrophil, that 40% more damage would be caused by the older neutrophil. But was this correct? We actually developed a way of measuring how much damage a neutrophil causes, and this is the measure here. And you can see when we compare an old and a young person's neutrophils, there's not a 40% difference. The damage is double. And this was associated with more inflammation. These are two markers in your blood, these cytokines that tell you how inflamed the blood is. So when you get any sort of infection, the older person has more inflammation, and it's that inflammation that causes the sickness and the disease, because it can damage your heart, your kidneys, your muscles, your brain. This is what does the damage. And it's even worse. The data I showed you were for healthy people. And here is where you get another difference with young people. So in the black bars here, these are young people. And this is the movement of their neutrophils and how efficient it is. These are healthy people. These are people who've got a lower respiratory tract infection, so a nasty chest infection. These are people with pneumonia, and the last group is sepsis. Hopefully you can see with the young people, when they get sick with a respiratory infection, a chest infection, their neutrophils actually behave and perform even better. We call this priming. The exception is sepsis. This is such a bad infection that even the young people, their neutrophils don't work well. But look at the older people in the white bars. There's the healthy, but it gets worse and worse. The more infection they've got, the more severe the infection, the neutrophils just don't move well. So how to understand this? And I know you are an audience that is interested in science, so I'm going to walk you through this rather horrible figure, but hopefully it's very clear. This is the neutrophil, and this is how it gets signals from that site of infection. Those cytokines land on the outside of the cell. They bound to these structures called receptors, and they send a signal through the cell. And the first signal is this thing called PI3 kinase. So we hypothesize that clearly what must be wrong is that this signal doesn't happen in older people's neutrophils. So we had to check this out. Now, this is a technique called Western blotting. It's a little bit like an X-ray. What we do is we give the cells a signal, and then we smash them up, and then we look at the proteins inside. And this is this PI3 kinase. This is control. This is freshly isolated neutrophils. And this line you can see, the darker it is, the more of that signal there is. And in the young person, when we give them these different signals, they all give a lovely dark signal. When we look at the older person, I hope you can see that there's no difference. The signal is the same the whole way across from the control value. But the surprising thing was that in the control, look here, that PI3 kinase is already active, whereas it isn't in the young people. So we had to rethink. We thought, well, perhaps what we've got to do is to inhibit that PI3 kinase, because it looks like it's active all the time in the older person. Perhaps that is the problem. And sure enough, we did this. We took these drugs, and there are 4 different forms of this PI3 kinase in the neutrophil: alpha, beta, gamma, and delta. And we found that if we inhibited either delta or gamma, that the neutrophils move better. They were just like a young person's neutrophils. So we thought, we've got an answer here. We've got potentially a way of improving the situation. But we couldn't use any of these drugs. They're all used in chemotherapy. They're really severe drugs. So we had to find another way. So we went downstream. So this PI3 kinase goes in this flow here. And we thought, well, let's see if we can inhibit this protein at the bottom. It's called a G protein. And what we knew was that drugs called statins, which you will all have heard of, these are drugs that you are given if your cholesterol is very high. And this is how they work. They inhibit this enzyme here called HMG-CoA reductase, which is required to make cholesterol. But it also causes the production of this protein here, which has got a horrible name, geranyl. But it's important for those G proteins. Without it, they can't function. So would statins do the same thing? Well, they did. These are the chemotaxis, that movement data for young people. This is without the statin, with the statin, and the young people don't have a problem anyway, so no great difference. These are the old people without the statin, and then with the statin, and you can see that their neutrophils moved just as well as a young person's. This is all in vitro, so in a test tube in the lab. And the effect was concentration dependent. The more statin we gave the neutrophil, the better it moved. But would it work in vivo in a real person? And would it work in a sick person? So this is a trial we did, giving simvastatin, one of the statins commonly used, to people with pneumonia, older patients. They all had pneumonia, and we gave half of the patients 80 milligrams daily of statin or a placebo, so a tablet with no drug in it, for just 7 days when they were in hospital. We looked at a whole range of their neutrophil functions, we looked at safety, and we looked at survival of the patients. And this is what happened. This is their chemotaxis. These are the patients on the placebo, essentially no difference. This is their, was it different from their control, their baseline value? But look at the ones on the simvastatin. After just 7 days, their neutrophils moved better. But more importantly, look here on the left, this is a score of how sick the patients were. And the ones on the statin essentially were better after they'd had the statin treatment. And when we followed the patients for a year afterwards, this is a survival curve. So each step is a patient passing away. And these are the ones who had the placebo. And these are the ones who had statin. So hopefully you can see that more of the statin patients survived. And in fact, it was 40% more survived. So the statin reduced death in these pneumonia patients. And again, in COVID-19, what many of the patients actually pass away from is they have the virus, but then they get pneumonia. So could the same be true for COVID-19? And a proper trial is only just starting. They're now giving statins to patients with COVID-19 in the UK. But these are some data from China, where they looked at patients who came into hospital with COVID-19 who were already using statins. And hopefully you can see these are the statin users on the top and the non-statin users. And this is how, this is their 28-day survival. So how many of them were alive 28 days later? And you can see that the statin users, more of them survived. And interestingly, when we looked at their inflammation that I told you about, this is the thing that the neutrophil does when it's not moving correctly, this was lower in the statin users. So the statin users survived better and they were less sick because they had this lower inflammation. So it's going to be interesting going forward to see how that statin trial performs. So I'm going to finish on a positive note. So the trials are ongoing with a whole range of drugs, but our best protection is really from vaccines. And as this is a German audience, I thought I had to focus on the Pfizer-Biontech vaccine, but the Moderna has a similar method. And how these vaccines work is what they do is they actually consist of that RNA that I told you about at the start that encodes for that spike protein, the SARS-CoV-2, it encodes for its material, but just the spike protein. So this gets taken up when it's injected into you, it's taken up into the cells in your body, and then your own cells make the spike protein and stick it on the surface. That then trains your immune system. You haven't really got the infection, you're just making that bit of the virus. So it's very safe. But it shows that spike protein to your immune system, and your clever bit of your immune system then makes antibodies and it makes T cells so that you're then protected. This is called immune memory. And when the vaccine trials were going on, most of us immunologists were very worried, because pretty well all vaccines don't work as well as you get older, because unfortunately that adaptive, the clever side of your immune system also doesn't tend to function well. These are data for the tetanus vaccination, and above the red line, people have made enough antibody to be protected. Below the red line, they're not protected. And essentially immediately after tetanus, if you're 70 years and younger, you're likely to be protected, but you can see the real decline with age. 70 years and older, you're not. And when you look 6 to 10 years later, it's really only the under 50-year-olds that have still got antibody. And the same is pretty well true for every vaccine like flu. So imagine the joy when the data came out that older adults did respond to the Pfizer vaccine. These are data from Birmingham, where I'm based. This was 100 subjects done by a colleague of mine, Helen Parry and Paul Moss. They were all aged over 80. The average age was 84. They were all care home residents, so not the fittest of 80-year-olds. And these are the data after their second dose of the Pfizer-Biontech vaccine. And you can see pretty well all, except for this person down here, pretty well all produced a really good level of antibody. And interestingly, the ones in the blue here, these are the ones who had already had COVID and survived it, and they were even better. And when we looked at their T cells, so those clever T cells that give you memory as well, the ones that already had the vaccine, the infection, did really well, as did most of the older adults. So that Pfizer-Biontech vaccine and pretty well all of the others work as well in older adults, which is a real relief. So while the virus itself can infect the older adults, it looks like the vaccine can protect them. So at that point, I'll finish, and thank you for your attention. And I hope that's really told you as much as you need to know about the vaccine and the COVID-19 disease.